of a very systematic study that we performed in order to really understand what kind of factors contribute to variations in the quantity of DNA purified from FFPE material. So I would start right away with just giving you an overview on the agenda and the various topics I will touch on. So I will start with a brief introduction about FFPE samples as a starting material for nucleic acid purification, trends and needs in the market space, as well as its challenges, of course, before quickly reviewing some studies which results have already been published, and then of course going into the specifics of what we have actually done here at QIAGEN in terms of what I mentioned before, understanding where the variation in quantity of DNA purified from FFPE materials come from, before of course finally then closing off with a summary. So I would say that within the research community, FFPE samples are not necessarily the most, let's say, favored starting material, as FFPE presents some challenges. And we're coming to that in a minute, what these are. But sometimes there is simply no other and no better choice. And the reason for that being that during the past decades, formalin fixation and paraffin embedding has become sort of a standard method for long-term preservation of tissue biopsies. And on the one hand side, in a lot of the cases, the choice here is not necessarily made by the laboratories performing the nucleic acid-based test, but by the personnel removing such biopsy samples. So for some of you, this means you simply have to deal with these samples because this is what you get in the end. But moreover, a large number of unprocessed FFPE samples are archived in tissue banks and biorepositories, representing, let's say, a highly valuable source, especially when these samples are well characterized. When it comes then to the needs of researchers working with FFPE samples, optimal and maximal recovery is especially important, since in a lot of cases these samples are very small and non-retrievable, I would say. And in the ideal world, DNA purified from such samples shall be usable for all types of downstream analysis technologies, including, of course, NGS, which also means co-purification of RNA shall be avoided whenever possible. And lastly, when DNA is the target molecule, it should be ensured that only true mutations are detected. So mutations originally present in the sample itself, and not artifacts that may have been created or formed due to the fixation and embedding or purification process in the end itself. So having said that, let's have a look at the important considerations when working with FFPE samples. And on this upper part here of the slide, so up here, you see the complete workflow from sample collection, actually, and handling to the molecular analysis of FFPE-based samples. And below that, in this portion here, I listed a number of factors that may have an influence on the final nucleic acid quality, and thus also in the end quality of your downstream results. And I don't even claim this list, honestly, to be complete. So just to mention a few examples here. Challenges potentially influencing your downstream results start as early as when handling, when the sample is taken, more or less. So the time between excision of a biopsy and fixation actually matters, as changes in the transcription profile and protein profile will occur during this time. But then also the fixation, the formalin fixation itself has an impact, as besides the fact that the chemical reactions taking place during the fixation usually lead to cross-linking between biomolecules, including cross-links between nucleic acids and proteins, and an acidic pH may cause hydrolytic nucleic acid fragmentation. But also three other factors are important and may influence your result, and this is and the extent of tissue fixation, which is the thickness of the tissue specimen, the volume of the formalin solution, and the duration of the fixation process. Underfixation can lead to nucleic acid and protein degradation, or a change in gene expression in deeper regions of the tissue that you're processing, and these regions that have not eventually been penetrated by the formalin. On the other side, overfixation can result in more extensive cross-linking, which makes extraction of usable nucleic acids and proteins more difficult. But then also paraffin embedding, as well as the final sample storage, may influence the sample quality and thus your final result. And with all of that being said, of course, also then the DNA purification process itself needs to be considered. So in terms of overcoming the challenges of DNA purification from FFPE samples, QIAGEN also offers solutions to that. For example, the QIAamp DNA FFPE tissue kit. And I mentioned that here with also the flow chart being shown, because that's exactly the kit that we used in our study that I'm going to describe. So on the slide, I wanted to provide some background information on the purification technology used and the kit specifications. So it's actually a silica membrane-based process that enables you to process up to 8 sections with a given recommendation on the thickness of that sample and the surface area. And it would allow you to purify genomic DNA as well as mitochondrial DNA with a variable elution volume between 20 and 100 microliters. And just to mention that there is also an automated protocol available here on the QIAcube system, and this is also something we applied in our study here. In terms of the paraffin removal and lysis of the samples, the procedure actually does not require any overnight incubation. The paraffin is dissolved in xylene and then removed, and the samples are subsequently lysed under denaturing conditions with a Prod K step, which helps to release the DNA by digesting cross-linked proteins. And this is followed by an incubation at 90 degrees for about an hour, and this incubation is done in an optimized buffer that partially reverses cross-linking. And during this incubation, methylene bridges between amino groups are broken, and formaldehyde molecules are released, binding to acceptor molecules. Although this excessive heating can lead to DNA degradation, I should state that studies at QIAGEN revealed that this is a necessary step and enables optimal recovery of analyzable genomic DNA. I should also say that in the kit protocol, the RNase treatment is given as an optional step, adding RNase to the sample and incubate at room temperature for 2 minutes prior to binding of the DNA to the spin column. So this is the general process. So having said that, I would like to take a brief minute to talk a bit about some prior publications. And please take those shown here only as examples. There are many more. And I would also like to mention that the purpose here is not to question the scientific relevance of these publications, but reading through these papers, we simply recognized that, yeah, we had a few questions here, and the messages provided to us seem to be a little, let's say, different sometimes. And for example, or to give an example here, in one study, for example, tissue types were not really specified. It simply said FFPE samples. It wasn't clear from what type of tissue. And also in some publications, it simply said DNA was purified according to the manufacturer's instructions, giving a kit and manufacturer name. But as I mentioned on the last slide, even in our QIAGEN protocols, something like an RNase digestion step is optional. So it wasn't necessarily clear, is that something that was performed or not performed? It was simply unclear. In some of these also in some of these papers that we found, also yields that were obtained were not given as absolute yields, but relative yields, which raises at least certain questions like what is exactly the amount, what is the technology that was used for quantification, and so on. So my main takeaway here is simply there were a lot of questions that we had, and we wanted to really investigate more or less what the title of this presentation assess where does variation in DNA quantities measured comes from. So to give you an overview of what we have actually done. So this is the study design. So we wanted to set up our own study to really systematically assess which factors contribute to the variations in DNA quantities measured from FFPE samples, and also ultimately to determine which factors do have the highest influence here. And what we did, we obtained red tissue samples from a cooperation partner. Actually, it was fresh tissue material that was fixed in formalin and transported at room temperature. After receiving these samples, we performed the same day dehydration and embedding in paraffin using an automated system, namely the Leica TP1020. And samples were subsequently stored -20 until further use. Of course, we recognize that this is sort of an ideal scenario and might not match your daily experience when you, for example, should receive pre-cut sections on slides. But for the purpose of the study, we chose to go down this route very simply. So what we then did, and we actually had 5 different red tissue types included here, and from these tissues we embedded in 2 to 3 different blocks, and we ultimately, from each of these 2 to 3 different blocks, we took 5 to 6 times 3 sections of 10 micrometer thickness to assess the variability caused by the samples themselves. We then purified DNA from these samples, either using the manual procedure or the automated protocol that is available on the QIAcube, using the kit that I mentioned, the QIAamp DNA FFPE kit, where we either did an RNase digest or did not do an RNase digest. So to assess the variability introduced by eventually operators also, looking at manual versus automated. And in the end, from the eluates, from the final eluates, we used those 3 methods given here, nanodrop, QIAGEN, and the qubit system, and measured 3 replicates of 5 replicates, of course, 5 replicates of each sample with each of the 3 methods shown here. So to assess the variability caused by the downstream quantification method. So in total, we're talking about 6,000 data points that were generated here. But before we go actually into the details of the results of the study, I would like to spend just a few words on the different nucleic acid quantitation technologies listed here that we also used in our experiment. So this table here provides an overview on the quantitation methods, and as you can see of them, can see 2 of them, namely actually the nanodrop and the QIAGEN, are based on absorbance measurement, while the qubit system is based on a fluorescent assay. So this assay contains a dye that only provides a light signal when bound to DNA, which means that only the molecule of interest, so DNA, is measured here, and no potential contaminants, including, for example, also residual RNA, which still might be in the sample, is measured using this technology. So before coming to the results of our FFPE study, I would like to just give you some background information from prior experiments that we did that I would like you actually to keep in mind when looking at the final study data. So what we have done previously to get a better feel for those 3 measurement technologies is we compared, we used a commercially available, as mentioned here, ultra pure gDNA with a given concentration, so this is the specified concentration, and measured this sample on all 3 systems. So, and this is the comparison from that here. And as you can see, all of the technologies, because the black bar here is the target concentration, all 3 technologies under-quantify actually the sample a little. But you can also see that at this point of 173 nanograms per microliter, the measurement points on both absorbance-based technologies are lying pretty nicely together, versus on the qubit there is a pretty wide variation of measurements. in terms of the under-quantification, of course, the magic question is, how was the initial concentration determined by Promega? So is it true that actually 173 nanograms are in these samples? But in terms of the variability, sort of you can clearly see that at this concentration range, the absorbance-based measurement gives the better reproducibility, I would say, versus the fluorescence-based measurement. The other thing to keep in mind is that, and we tested that, we diluted this ultra-pure gDNA down to 5 nanogram per microliter. And when we did that and measured again, on all those 3 different systems, what you can see is that actually the variation on the nanodrop specifically, but also a little on the QIAxpert gets higher. But on the qubit system, all data points are lying very nicely together here. So this is just something to be aware of and keep in the back of the hat, I would say. And what that means when looking actually at the linearity of measurement over a broader range of concentrations is actually shown on this slide here. So what we noticed is, so you see a target concentration versus the actually measured concentration and 2 lines. Specifically here you see a black line and the red line where in the ideal case all data points would lie on the black line. But you can see that, that there are certain trends, I would say, in terms of that we see a slight systematic over-quantification using the nanodrop system here, and in our hands on the qubit system we saw a slight under-quantification. And this is just something I wanted you to be aware of. And going back to our study, I will now present the results of that study, starting from a very high level and subsequently drilling down into the details. So, so this is the first slide, and what we are looking here are actually all 6,000 data points measured. And what you will immediately notice is, compared to the qubit system over here, you see a much higher variability in the data points on both absorbance-based measurement systems. And this, of course, immediately triggers the question, why is that the case? What are the factors contributing to this variability when doing absorbance readings? So having in mind that that on the qubit system, based on the fluorescent assays used, only DNA is quantitated, we first looked at, for difference generated by samples treated with RNase versus samples not having been treated with RNase. And if you split out these data points by color, what we did on this slide here, and you see now all the data points from the samples that were not treated with DNase in blue versus the data points from the samples that were treated with RNase in red. You can immediately see that the majority of the variation is contributed by residual RNA, I would say, that was co-purified in those samples that have not been treated with RNase. And when you look at the table here, you can also see that on the QIAxpert, with the average value that we obtained, we got much closer to what actually we also measured with the qubit system, where we still see a higher variation in terms of measurement on the nanodrop. So the next question we asked then is, what about automated versus manual processing? Does that have an effect? And this is what we actually see on that slide here. So when we split out data points based on that, so we're looking at samples that were treated or that were purified on the QIAcube with an automated procedure versus manually. Yeah, so you can see, and if you just look at the red data points, because let's forget about the blue ones for now, you can see that overall there are less outliers here when you process samples in an automated way, and this is very obvious when we measure with a nanodrop here, so less outliers, but still some on the nanodrop measurement versus the manual procedure where we do see a bit more of outliers. And what we think this tells us is that, yeah, using an automated procedure for the purification simply provides a higher degree of standardization and therefore, yeah, a little improvement on your results, I would say. Now, you may still ask, because I said we used 5 different tissue types, so you still may ask a question, what role does actually the tissue type play here in the variation that we do see? And surely we also looked into that. And this is what you see on the next slide. So these box plots here were generated based on actually all data points we had. So taking all different measurement technologies into account. And yes, we do see differences based on the tissue type, where I should say not unexpectedly, red liver here shows the highest variation simply due to the fact that liver contains a lot of RNA, and this, as this is for all data points that we generated, also samples are included here that were not treated with RNase, so where RNA was potentially co-purified. So you may then say, okay, what then about RNase versus non-RNase treatment and in the different tissue types? So we also looked at that, and when you it becomes very obvious when you do so, and when you look at these box plots here, that again the majority of the variation on the absorbance-based measurements comes from measuring additional RNA, which is still in the samples. However, still, if you look at the red data points, I would say, and this is very obvious here with red liver, when measuring those samples, even those that were treated with RNase, so no RNA in there, a higher level of variability was still seen on the nanodrop, where versus, for example, here this data point or this data point on the QIAGEN versus the qubit system. So main contribution to variability related to the RNA amounts, but we of course drilled down further, sort of also looking, does the automation play a role here? The automated purification procedure, does it get better then? And just let me quickly go back, because one thing I would like to alert you to is this here. If you look at the scale here in terms of the maximum value, on this slide we're looking at a maximum of 850 nanograms per microliter. When you now only look at the samples that were purified with the automated procedure, on this slide here, the maximum is 360. So the variation is definitely lower using an automated procedure. But still, you see a similar picture as on the slide before. Major variation contributed by residual RNA. But if you look at still liver here, same picture in terms of even with RNase treatment, you see differences in sort of the variability based on the quantitation method that was actually used. So we drilled even further down then, saying, wanting to investigate what is the contribution to this overall variability then in the end, contributed by the block or even the section itself. And this is also something what we looked at. And in this case, we only we only display here the liver samples here. So this data set shown represents the liver samples only, and it represents the DNA quantities measured from the 5 sections taking from each block. So 1 to 3 in this case here means what was the FFPE block, and the dots here represent all the different sections that were taken from this block 1 versus block 2 versus block 3. And again, of course, we see the difference between samples purified with versus without RNase treatment, but overall, the difference in quantitation caused by the measurement system is higher compared to the difference between samples which were taken from the same block. So if you look at the different blocks, the picture looks more uniform for each of these measurement methods compared when you relative to a comparison looking at these measurements data versus these measurements data versus these measurements data. So higher differences in the yields measured in the end due to the quantification method chosen rather than the sample block. And finally, then yeah, we drilled down to the actual sections. We looked at the differences that could potentially be attributed by the individual sections that were processed. So what you see here on this slide is data from the 5 first sections that were taken from each of the 3 blocks. So 1 means these are the 5 first cuts from block 1, block 2, block 3, plotted together, and so on. And again, we see that the contribution to the differences in yields measured from the quantitation method is higher compared to the contribution coming from the sections themselves. And the nanodrop still shows the highest level of variability even after RNA digest, which is to us a little questionable and definitely something to consider. So having this huge data set at hand, we of course then wanted to determine, of all those factors we looked at, what is the factor having the highest influence on the quantity of DNA measured from FFPE samples. So we used a statistical software from the SAS Institute, which is named JUMP11, to analyze this. And the decision tree analysis method was used to determine which of the parameters evaluated has the greatest influence on the variation of DNA quantities measured. And the results of this analysis show that in our study, what mattered most was actually, and this is shown in both in the table and also in this bar graph here, is what matters most was the quantification technology chosen, contributing with 28% to the overall variability, followed then by what is called the purification method here, so whether an automated process was chosen or samples were processed manually, and only then by tissue type, which is still 23%, I would say, the RNA to digest. But what was very, very clear is what matters least is actually the FFPE block or the FFPE section here. So chosen quantification technology, at least in our study here, mattered most. And to sum this up, what we really recognized and feel is, yes, FFPE tissue samples present a number of challenges. And if we would need to make a recommendation based on the study we did, we would say, if you really want to be sure that the genomic DNA you quantify represents what is actually in your sample, first thing is choose your quantification technology carefully. Be aware of what differences might be. Definitely, we feel automate your sample prep because we could see that this provides a higher level of standardization and improvement, therefore standardization of results. And definitely apply an RNA digestion step because you saw the effects of what happens if you don't do that. So it's definitely something which we feel is mandatory here. And also we would say be aware of systematic differences between quantitation technologies chosen when measuring your DNA amounts. And what we also feel that we can see in this study that the QIAGEN system really offers a nice and reliable quantification of FFPE samples and is a good alternative, I would say, also to the qubit system that a lot of you may use today. So overall, yeah, QIAGEN provides a number of solutions from sample to insight, supporting your research efforts using FFPE samples. And with that being said, I would like to open up, I think there's one more slide, saying thanks for listening, and I'm happy to take any questions.
Great presentation, thank you very much. If you have any questions, please feel free to type them in your questions box. I'll start the first question asked by Charlene, and ask any reason why nanodrop has higher variability than the other 2 systems.
This is, of course, it's hard to tell. Let's say it's an observation that we made here in our case, that we found this higher variability. What is exactly the reason, I would say, is speculation from our end. It might be this is speculation, as I said. So of course, on a nanodrop system, you need to make sure that this liquid column is really nicely built up. We feel we handle the system well and have it under control, but still we saw this variability. And of course, it's a big question: where does this come from? And it's a question I think researchers should and will ask themselves. So it's really making sure use your nanodrop, try to see how reproducible results does it give to you, and I'm afraid that's all we can say here.
Great, thank you. Another question also asked by Charlene. I'm not sure if you are able to, give some insight. Would you comment on the gene read product? That's for the NGS panels.
Yeah. So honestly, this is something we only realized a little later. We probably could have extended our study to also look at how do results look like when we use the, let's say, standard QIAM DNA FFPE kit versus the gene read DNA FFPE kit. But for this study, I have to say we were a little late in recognizing that. So we were not able to include that later on. So this is definitely something we would like to look into. Also, maybe in a follow-up study and experiment, but I have to say we haven't done it yet.
Thank you. Another question asked about how you used HS or BR credit. When
I look at the range of concentrations, I'm pretty sure it was the standard BR kit, so not the high sensitivity kit, in order to cover the entire range. But I'm also, and I can check on that, but just from the range of concentrations that were in this presentation here, that's what I would say.
Great. Yeah, some of you asked about the handouts, the webinar slides and the recording. Yes, after this webinar, everyone will receive a copy of webinar slides and also the recording link. And here I got another technical question. Is it possible to do specific macron expression analysis in FFPE sections on glass slide?
I didn't get the question fully. Is it possible to use what?
To do the macron expression analysis. From the FFPE section on glass
slides. So
yeah, glass
purifying, so doing microarray as a downstream from samples that came from a glass slide. I would say yes, but maybe where you can capture the contact, because we can check on that, whether we have some in-house data here.
Okay, great. Got another question related to NGS. Do you have any experience with NGS?
That's a very generic question. Personally, I have to say no. Within the company, definitely yes. So in terms of downstream analysis technology being used. If whoever asked this question could be a little more specific and what kind of information you would like to get, I'm pretty sure we can capture this information and try to get back to you on that one.
Great. Another really also similar question about the NGS. Does the variation in quantity of DNA in FFPE sections can affect NGS results?
This is also something we still would like to determine, because of course it's a long way in terms of workflow and process from the actual primary sample to final library and NGS results. However, what we feel is that of course a misquantification after this first step also means that you are either putting a higher or lower amount of DNA in your next step of the workflow, and what kind of impact that finally has on the next, first of all on the next step, and then finally does that matter in the final NGS result is still something that we would need to look into. We haven't done any experiments in-house on that yet, but we're definitely planning within looking at the overall NGS workflow. We definitely use all the methods that I showed to you here in terms of quantification, and this is definitely something we should get more information as we go along.
Great, thank you. I get another very positive feedback from Matthew, and he said a very nice summary of QIAGEN products related to measuring quantities of DNA. Thank you, Matthew. I'm glad you found this very nice. And he asked, is this part of this presentation about FFPE about to start? So I don't quite understand, I guess. He's asking, is this the first presentation we have about FFPE, or later on are we going to have more webinars? Is that what you're asking? Okay. What do you think, Dr.
Yeah, so let's say there would be different aspects. FFPE is definitely an important topic, and we can look for it. I think we did other FFPE-related webinars, so we can check on what information we have there. When it comes to quality control of such samples, because I gave this webinar before, and there were questions like, did you also look at RNA from FFPE, for example, questions like this. And I can tell you, last time I had to say we're still working on it. This time I can say, yes, we have done a similar study also looking at RNA from FFPE material. The data is available, but it's not yet put in such a nice presentation like this one here, but we're working on it, and we're happy to keep you up to date with these results in a future webinar on that.
That's great, thank you. Another question is about, again, FFPE on real-time PCR. So what's the most important impact of the extracted DNA from FFPE tissues on real-time PCR?
Oops. That's a hard question, because as I showed in the beginning, there are so many things that might have an influence. I would like, whoever asked this, we have a very comprehensive, actually, brochure on FFPE samples and impact of different parameters on downstream results. And I'm pretty sure we're happy to share this information with you, as it's already in a nice brochure format, because that gives you a very comprehensive overview on what is the impact of certain things like overfixation, underfixation, purification, and so on downstream results. And I would say let's get this brochure to you for a read up, and if you should have any follow-up questions, I'm pretty sure we're happy to discuss that with you.
Yeah, thank you. I'll make sure everyone receive a copy of that brochure. And we have another question about actually bacteria DNA. Have you ever tried to verify bacteria DNA isolation from FFPE samples?
This is something I would need to check with R&D here, actually. I'm not aware of it currently, but I'm not saying nobody has tried it in-house. So this is something we definitely need to check with the R&D department.
Okay, great. Thank you. Another question about histology samples and compared to tissue blocks. Sean actually asked this question. Do you have any experience with macro dissection sections from histology slides?
Yeah, I'm pretty sure we have. In terms of the purification process, again, I would say it would be nice to know what piece of information you exactly would be after. But I'm also aware of we have a number of customers actually doing that. So it would be nice to know what information exactly the person who asked that is after, and I'm pretty sure we could try to gather that.
Great. I think that's all the questions I have. And
just a question from my side, where the questions will be collected also with the person who asked, so we could get back, right?
Yes.
Perfect.
Yes. And also the brochure, I can see we receive a lot of questions regarding the downstream analysis, you know, the impact of FFPE on the downstream, like a qPCR, NGS, and macro profiling. I believe the brochure you just mentioned covers all these, so I'll make sure everyone receive a copy of this brochure. Okay. Any other you would like to talk about more? So should I close the session? Okay. Okay. So if there are no more questions, I'll close the session. Thank you everyone for attending this webinar, and also thank you very much, Dr. Kavula Shard, to give this very nice presentation, and wish you good luck in your research. Thank you. Bye-bye.
Thank you. Bye.